Hey Satterton, this is Mr. Dubik, and in this YouTube video we will be talking about DNA replication. DNA replication is the process by which DNA makes a copy of itself. Why is that important? Well, if you look at this image to the right, you can see the sperm cell is being fertilized by the egg. That means it's about to become a zygote, which is the first cell of a new child, which you are seeing right here. That zygote will then go and divide through a process called mitosis, which will eventually become an embryo, okay, which we can see the growth and development of a new human. And then it will be eventually turning into a fetus, and then will eventually create a new human. And that human is going to be made up of millions and millions of cells, just like you are today. We have to make sure that each one of those cells has the same exact DNA that was in the first original cell called the zygote. And we do that through the process called DNA replication. So here we go. So on this slide, what we're going to talk about is where and when does DNA replication occur. And if you look right here, you can see the cell cycle, and this is the cycle our cells take in order to divide, meaning going from one cell to two identical cells so that we essentially can grow and develop. So DNA replication occurs during the S phase in the cell cycle, which is down here. S means and stands for synthesis, which makes sense, okay? And the S phase equals synthesize, which means to make. So therefore, our cells are making DNA right here in the S phase during cell division, okay? And we call this process right here mitosis. Okay, so DNA replication is often referred to as a semi-conservative process. And why is it often referred to as a semi-conservative process? Is because when we want to create two new identical DNA molecules, okay, from the original DNA molecule, what we're going to have to do is we're going to have to split apart the original DNA molecule into two DNA strands. Those strands are going to act as a template strand to allow two new strands to bond complementary to the original strands to essentially create two identical DNA molecules which are original of the original DNA molecule. So what is DNA and how is it a semi-conservative process? So during DNA replication, half of the original DNA molecule will be conserved during DNA replication to make two DNA molecules. Each original or old DNA strand will act as a template strand, allowing the complementary or new strand to be made. As a result of this process, what we get is we get two identical DNA molecules consisting of one old strand and one new strand. Okay, so now what we're going to do is we're going to use this diagram right here to explain what we just talked about. So this diagram to the right that I'm circling is the semi-conservative DNA replication process in three simple steps. The first step, what we have here is we have our original DNA molecule that needs to be split in order to make two new DNA molecules. Stage two is what we have here is we have two DNA strands, okay, and these DNA strands were split down the middle by an enzyme called helicase, okay. So now that we have two DNA strands, what we can then do is we can add the complementary nucleotides, okay, to these DNA strands to simply form two DNA molecules, and that's step three. So once the enzyme DNA polymerase added the complementary nucleotides, what we then form is we form two identical DNA molecules that are identical to the original DNA molecule. Again, that is the goal of DNA replication. We start with one original DNA molecule, split it open, add the complementary nucleotides to then form two new identical DNA molecules that are identical to the original. These DNA molecules will then go into two cells, okay? Each cell, therefore, will be then identical because their DNA is identical. And that is the purpose of DNA replication, to go from one original DNA molecule to two new identical DNA molecules. In this diagram right here, what you're seeing is you're seeing two DNA strands, and what again then you're seeing is you're seeing these nucleotides bonding complementary to the original or template strand to form two new DNA molecules. So again, DNA is a semi-conservative process. Okay, so on this slide what we're going to talk about is we're going to look at and go over the actual process of DNA replication in multiple steps. Okay, so what you're looking at here is you're looking at a DNA molecule being split apart and then replicated into two new DNA molecules. Here's one DNA molecule being formed, here's the other DNA molecule formed. Here is the original molecule being split apart. So step one, what we need to do in order to create two new identical DNA molecules is we need to split apart the original DNA molecule apart. So how do we do that? What we're going to do is we're going to use this enzyme called helicase, and helicase is going to simply go down the center of the original DNA molecule, and it's going to essentially hold together the two DNA strands. So as DNA helicase goes down the center of the original DNA molecule, it's breaking the hydrogen bonds that hold the DNA molecule together into two DNA strands. That is step one. And again, an easy way that I think of helicase is I think of helicase as a Pac-Man, because in the game of Pac-Man, Pac-Man simply moves around and begins to chomp at the dots, okay, so that he can get more points. So that's kind of what helicase does. It's chomping away at the hydrogen bonds, and it's essentially splitting apart the DNA molecule into two DNA strands. So once the DNA molecule begins to split apart, we're going to have these single-stranded binding proteins that are going to act as thumbtacks, and essentially what they're going to do is they're going to hold in place the two DNA strands that were zipped. Okay, and the reason for that is because when we begin to add the complementary base pairs to the two new strands, we have these single-stranded binding proteins that are holding those two DNA strands in place to make it really smooth for that DNA polymerase to add those complementary base pairs. So after the single-stranded binding proteins have been put into place and they're holding down the two DNA strands, what's going to then happen is DNA polymerase, which is an enzyme, is going to move down the two DNA strands, and it's going to add the complementary base pairs to the leading strand in a 5' to 3' direction. Okay, so anytime we're adding nucleotides or bases to the unpaired DNA strand, the DNA polymerase is always going to add the complementary base pairs in a 5' to 3' direction. Okay, and that's what you're seeing here in the leading strand. If you follow this 5', it starts here, and then the DNA polymerase is going to move down in the 3' direction, and it's going to add the complementary base pairs to form a new DNA molecule. So what does the DNA polymerase do? It simply will travel down the DNA strand, and it will add complementary base pairs to the leading strand in a 5' to a 3' direction. Now, where does this DNA polymerase know where to start to begin adding these complementary base pairs? That's where DNA primase comes in, and DNA primase is going to simply bond to a certain or a specific location on the DNA strand, and it's going to add an RNA primer, and that RNA primer is going to essentially signal to the DNA polymerase that is the location where it needs to start adding the complementary nucleotides. Okay, so once this DNA polymerase moves and travels all the way down, okay, the DNA strands and adds the complementary base pairs, what's then going to happen is DNA ligase is going to come in, and essentially what it's going to do is it's going to act as a cleanup crew. And then it will join the fragments together to complete the sugar to phosphate backbone, and it is also going to look for mistakes within the new DNA molecule, because again, there could be complementary base pairs that were bonded incorrectly. So therefore, that's why we have DNA ligase, who's going to act as a cleanup crew. It's going to make sure that the backbone phosphates is all put together, okay, so that there's no missing parts. Also, it's going to look for incorrect base pairs so that it can correct them, so that this DNA molecule can be complete and can go into a new cell. Okay, so those are the stages in DNA replication, and again, the whole purpose of DNA replication is to start with one original DNA molecule here, use helicase to travel down the center of it. It's going to unwind the original DNA molecule into two DNA strands. DNA primase is going to go ahead and it's going to add a RNA primer to the DNA strand. That's going to signal to DNA polymerase that that is where it needs to start adding complementary base pairs to then begin to form a new DNA molecule. And again, DNA polymerase always adds the complementary base pairs in a 5' to 3' direction. Okay, and then once the DNA polymerase has added all the complementary nucleotides, DNA ligase is just going to come into the DNA molecule. It's going to look for any incomplete base pairs. Also, it's going to make sure that the backbone, the sugar phosphate backbone, is correct and there's nothing missing. Once it's then complete, okay, we form 2 new identical DNA molecules that are original or identical to the starting DNA molecule. And again, that is the DNA replication process. Okay, so let's talk about DNA replication in an overview perspective. So the products of DNA replication are 2 identical DNA molecules. The 2 new DNA molecules are identical to the parent DNA molecule. Each molecule contains 1 original strain and one new strain of DNA, and we call that a semi-conservative process. So if you look here at, again, the overview of DNA replication in this diagram right here, you have your original DNA molecule. It's going to begin to split apart. Then what we're going to see is the new strand binding complementary to the original template strand. As a result, what we get is we get 2 new identical DNA molecules. Each DNA molecule will go into a new cell so that all the cells in whatever organism these DNAs are for, okay, can all be the same and they can all function properly. So again, that is the overview of DNA replication, and these are the products. Okay, so these are some concept questions. Okay, make sure you go ahead and answer these on your own. That is all for this video. I will also be posting a YouTube video that kind of demonstrates the actual process of DNA replication so that you really have a good understanding of how DNA is replicated and the enzymes that play a huge role in producing 2 new DNA molecules.